Okay, hi everyone. So welcome to another webinar as part of our Clinician Engineer Hub webinar series. I'm very excited to have Prof. Cesar de la Fuente here live from Pennsylvania in the United States. He's done an immense amount of work on the utilization of machines and biology to detect, prevent, and treat disease. And he has significant expertise in this particular topic as well, which he's going to talk to us about this afternoon. And that's on the utilization of artificial intelligence approaches for antibiotic discovery. So myself as a clinician, always prescribing antibiotics, I classify myself as a pseudo engineer, but definitely this is the future. So very grateful for Professor Cesar to join us. And I'm going to hand it over to him now and look forward to his presentation.
Great. Thank you, Neel, for the very kind introduction. It's really great to be here. So I thought what I would do today is share with the audience a little bit about the progress that we've made around utilizing artificial intelligence approaches to be able to discover novel antibiotics. And a lot of the audience is probably familiar, but developing drugs is extremely slow and expensive. So on average, from the moment that you discover something new in the laboratory to the moment that it has an impact on patients, it takes over 10 years on average. And it's an incredibly costly endeavor. So to develop a single drug, it costs around really over $2 billion US. And just to put this into perspective, this is more is more than the budget that NASA has to take a rocket all the way to the moon. So it's actually more costly to develop a drug than to fly in outer space. And in my lab at the University of Pennsylvania, what we think is that we can take advantage of the two trends that we've been seeing for the last decades. The first one is our ever increasing compute power. This is provided by the number of transistors that we can fit into a chip that has allowed computers to be able to process data to almost double their processing power every 2 years or so, following Moore's law. And in addition to that, we've also made a lot of progress in recent years in being able to generate data at a scale that is unprecedented. And this is due to advances in automation and data, high throughput data assays. And so we think that by taking advantage of our increasing compute power and our ability to generate a lot of data, we think that we can really help bridge the gap of the amount of time that it takes to develop novel drugs, and the amount of money that it's associated with discovering novel drugs. And in particular, we focus on trying to develop novel antibiotics. And the reason for this, the huge motivation behind this, is that antibiotic resistance is a global health problem. It affects every corner of the world. It's the current projection is that by the year 2050, around 10 million people will die every single year as a consequence of drug-resistant infections. We can see here that this will surpass every other major cause of death in our society, even including cancer. And essentially, those 10 million deaths per year, they translate, if you do a quick calculation, in about one human death every 3 seconds. So this is the future that we're heading towards, unless we are able to come up with novel types of antibiotics that are effective. And at this point in the talk, I always like to emphasize that antibiotics are not only helpful to treat an infection. When we have an infection, we take an antibiotic and we get cured. But they're also essential for modern medicine. And so for things like organ transplantation, you need effective antibiotics. For things like surgeries, you also need these drugs. Even like a chemotherapy treatment, cancer patients are oftentimes immunosuppressed, and so they're very susceptible to contracting infections. And so you actually need viable antibiotics as well. If you think about childbirth, even. So modern medicine would not be possible without these very essential drugs. And so that's kind of like a huge motivator for my team to go after this big problem. And the problem that we're facing is that the biological world, which has been really a great inspiration of novel antibiotics. So basically, from the very first antibiotic, which was penicillin, which was discovered by Alexander Fleming in 1928, almost every other antibiotic that we've discovered in the world has actually come from nature. The problem that we're facing today is that it seems like the biological world, like I say on this slide, is running out of antibiotic chemistries. We've been unable to find any novel antibiotics in the last past couple of decades, and this has, and here I'm illustrating some of the chemistries that the biological world has provided us with, and that have been allow us to save millions of lives around the world. But again, like I said, now we're really running out of new chemistries in nature. And so in my lab, what we're thinking is, instead of relying on nature as a source of inspiration for novel antibiotic drugs, why not try to encode this chemical complexity and translate it into the binary code of ones and zeros that computers understand, so then machines can take care of the discovery process. And the way we typically do the engineering, and in my lab we focus mostly on proteins and small proteins called peptides, is that traditionally, essentially, we follow rules that tell us that the structure of a protein or a peptide determines its function. And so basically this means that if we can control the sequence of the building blocks of proteins and peptides, which are called amino acids, we'll be able to control their function. And this is a little bit like playing Lego. We're trying to rearrange the building blocks in specific positions to be able to create novel antibiotics that work. And the framework that we use in the lab is that we first want to understand these small peptides that we work with from basic principles, meaning the underlying physics and chemistry that dictates the biological function of these particular molecules. Once we've reached that level of understanding, we want to be able to control them. We do this through different means, including chemistry and biology. And once we're able to control them in the laboratory, we want to digitize all that information. So then computers, like I mentioned, can accelerate the process of discovery. For today's talk, I'll focus on this, the latter approach, how we computerize a lot of this information. And computers can help us do many things in drug discovery, but for the purpose of today's talk, I'll focus on three major areas. The first one is that they can help us explore sequence space. This, as you'll see, can be very helpful. They can also enable us to generate new molecules, and here we enter into the realm of creativity, right? Creating new molecules, and which creativity is a term that we typically associate with the human brain, but I'll show you some early advances in teaching machines how to create new things as well. And finally, they can help us mine biology because they can process huge throughputs of data, and they can allow us to find interesting patterns in biological systems. So I'll start with the first one, exploring sequence space. And the sequence space of proteins and peptides is incredibly immense. To illustrate its vastness, I'm showing here the combinatorial spaces of concepts that perhaps are more familiar to the audience. For example, number of people on Earth is around 10 to the 10. The number of stars in the universe, 10 to the 31. Now, let's consider a very small protein, a peptide, composed of only 25 amino acids, 25 building blocks next to each other on a linear chain, a little bit like a collar of pearls. Well, that gives us a sequence space that is actually superior to the number of stars in the universe. And that's, again, a very small peptide, smaller than any protein that is in our bodies, which are typically composed of hundreds of amino acids. And so we clearly need computers to be able to explore this incredibly expansive sequence space. And to make things even more complicated and therefore more interesting from a scientific perspective, biological evolution through billions of years has only sampled a tiny fraction of all potential molecules or proteins or peptides. And in fact, we can fit them all in this little pink oval here. And all this white space has never been explored. All this molecular sequence space has never been explored throughout the evolutionary process. And so what we think computers can help us do is expand the boundaries of sequence space to be able to reach some of these wide areas here that may yield novel drugs, in our case, novel antibiotics that may help us clear infections that are currently untreatable. And so the next question that we ask the team is, how can we clearly need computers to solve a lot of these problems that I'm posing, but how can we innovate at the molecular level? How can we create diversity at the molecular level? And after thinking about this for a while, we decided that the best solution here was to actually mimic the greatest engine we have for innovation, and that is no other than evolution itself, right? Through evolution, that's how organisms evolve and how innovation is created throughout time. And so we decided simply to mimic Darwin's algorithm of evolution and to be able to train computers to execute this algorithm. And so this is how we did it. We started with an initial population of peptide sequences from nature, and then we taught the computer how to perform evolution. So the essential steps are mutation, selection, recombination, and then n-fold iterations of that feedback loop, and that essentially creates evolution on the computer. So here we can see an example where upon increasing number of iterations, the computer is able to evolve the molecules towards an optimal fitness values that correspond with predicted antimicrobial activity. So essentially, the computer's making the antibiotics better. These peptide sequences, it's turning them into better antibiotics, at least in principle. And in the process of doing that, it's actually exploring previously unexplored regions of sequence space, the wide areas that I mentioned 2 slides ago, and giving us sequences with amino acid ratios that are actually atypical, not the typical things that we see in nature that evolution has created. And so really the machine is capable of creating things that look new and unprecedented. So what we see here on the screen is a bunch of different peptides that the machine created. Again, up to this point with very, very minimal human intervention. And we can see the alpha helical structures, and this is all really nice. But of course, here we reach a roadblock that is common to any project involving computers, right? And that is that the computer, everything we see is based on computational assumptions. The computer is thinking that these molecules would be great antibiotics, but we're not at a stage in this field where we can just completely trust the computer's assumptions. And so we need to validate everything. And this is where it comes really handy to not only do computational work, but also experimental work in the laboratory. And in our lab, we're lucky that we can do both. And so what we did here is we synthesized a lot of these peptides here using chemistry, solid phase chemical synthesis, and then we tested them against real bacteria that are clinically relevant. And we were able to identify through those screens a lead molecule that was very powerful, a very powerful antibiotic. And here's that lead molecule. This is the three-dimensional structure as elucidated by nuclear magnetic resonance. We can see again this beautiful alpha helical conformation. And this is the same molecule that we call Wavanin 2 in 3D space. Of course, with our collaborators, we were not just happy with this. We wanted to see if this machine-made molecule was capable of blocking infections in a preclinical mouse model, right? Because we're interested in translating some of these technologies to the next phases. And so we developed this mouse model. And here we can see remarkably that the computationally generated molecule reduced the infections a lot better than 2 of the template peptides that the computer started the optimization process with. And here we have the untreated group of mice, so you get a sense of how many bacteria, around 10 to the 7 bacteria are present in those untreated mice. So it's quite a lot of, they're quite infected, and really the synthetic peptide that we generated was capable of really reducing the infection very, very significantly. So basically, I've told you how we can now we enter a new phase in the field of AI for antibiotic discovery, where we can design things on the computer that are not only interesting conceptually, but we can then synthesize those molecules, and they actually are able to kill bacteria, real bacteria that kill people. They're able to kill them in the laboratory, in vitro, but also in preclinical mouse models. This is another project where we identified a pentapeptide motif indicated by this red color. So this sequence here that was associated with two functions: antimicrobial activity, so direct antimicrobial activity, and then immunomodulatory activity. And so the hypothesis that we had here is that, can we stitch this sequence onto a different sequence to endow that other sequence these two functions that we're interested in, so antimicrobial and immunomodulatory. So a little bit like playing Lego. And that's what we did here. We took this toxin that has this sequence code of amino acids, and we engineered onto its N terminus the pentapeptide motif here. So in the end, it's giving us the synthetic molecule that has the pentapeptide motif in red stitched onto the normal sequence of the toxin. And this was a really fun project in collaboration, where we were able to demonstrate that the synthetic molecule was actually capable of both properties. It was capable of killing bacteria directly through its antimicrobial activity, and it was capable of boosting the host's own immune system to clear the infection through its immunomodulatory properties. And we're able to demonstrate this in vitro in a skin infection model, and even here in a sepsis infection model, where we saw that treatment with the synthetic peptide conferred protection and against otherwise lethal infections caused by both laboratory strains and the B and D, and also by drug-resistant strains from the hospital. So also highly clinically relevant bacteria from the hospital. And this was quite fascinating to see that, the treatment was actually able to confer a lot of protection to the mice against, infections that are typically very lethal very fast. Okay, so far I've told you how we can use computers to design novel types of antibiotics, and in this next part of the talk, I'd like to tell you how we can use computers and computational tools to be able to discover new antibiotics in biology specifically. And biological information has a lot of information, a lot of code that has not really been explored properly. When we first sequenced the human genome and all the other subsequent genomes, we identified the concept of junk DNA, that we thought junk DNA was not really doing anything. But of course, now we understand that it is doing a lot of it has a lot of functional roles. And similarly, there are a lot of other regions in genomes and proteomes that have really not been explored as a source of potential drugs, of potential antibiotics. And to tackle this problem, we took inspiration from image and speech recognition algorithms that are used, every day, right, on our phones, Siri, Alexa, and so on. But here, instead of recognizing facial expressions or sounds, we wanted to recognize molecular patterns that were associated with antibiotic activity. So a little bit, the analogy here would be that, let's say if you had a huge Word document, let's say a PhD thesis of hundreds of pages, and you wanted to find within that document a particular word, let's say biotechnology. You would go to the search function in Word, you would type biotechnology, and the function would find the word biotechnology in every instance that it appears within the document. So these algorithms operate a little bit like the search function. We kind of know the patterns that we're looking for, and we can go to the search function, we can type them in, and then it finds them in the document. And the documents that we use to explore are not PhD thesis or a particular Word document, but we use entire genomes and proteomes. So this really allows you to do proteome-wide and genome-wide explorations to try to find novel antibiotics. And to illustrate this concept a little bit more visually, this is kind of how it works. So if you have a protein in 3 dimensions, you can display it in 2 dimensions with the amino acid code. And the algorithm essentially runs through the code and it identifies regions that are predicted to be antibiotic. And this is given by these different colors here. And again, this is just a prediction, but it's a very powerful prediction because it allows you to identify regions within entire proteins, such as this region in yellow, that may constitute a potential antimicrobial molecule. And then we can take that fragment out of the entire protein and play around with it in the laboratory to see if it has some sort of activity. We've essentially employed this method to explore the entire human proteome for the first time as a source of antibiotics. And the really fascinating thing from our perspective is that we were able to find antibiotic molecules that we call encrypted peptides, not only in the innate immune system, which is where you would envision you would find antimicrobial molecules, right? The innate immune system is the one that, when an invading pathogen enters our bodies, is the one that mounts an immune response that allows us to fight off that particular infection. But we also found them, in addition to the innate immune system, we found them in the cardiovascular system, nervous system, digestive system, all around the body. And this has taken us to a current hypothesis that we have now in the lab, that is that perhaps immunological response is not only a responsibility of the innate immune system, but we have all these other body systems cooperating either actively or passively to host defense. So I'll tell you briefly some interesting properties that we found from these encrypted peptides. So essentially, we identified thousands in a human body, and we were able to synthesize 56 of them using chemistry to test in the laboratory. And one interesting property that we found is that if we took sequences that are produced within the same area of the body, let's say the bloodstream, and you combine them in cocktails, they were able to synergize with each other to potentiate each other's activity to target pathogens. I thought that was a really interesting observation. And that's what is showing here in a rather complicated experiment. And the other property that I wanted to tell you about is that obviously when you're trying to develop a potential antimicrobial lead, or any drug really, you don't want the target to develop resistance to the drug. And that's why we perform antimicrobial resistance assays, where we expose bacteria, in this case Acinetobacter baumannii, a rather recalcitrant pathogen that is really difficult to treat. Actually, we expose it continuously to a conventional antibiotic that we use as control, in this case polymyxin B. This is an antibiotic that is used every day in the hospital. And then 3 of our encrypted peptides from the human body. And we expose the bacterium every single day to these agents, and we can see that eventually the pathogen develops resistance to the conventional antibiotic, but it doesn't really develop resistance to the encrypted antibiotics that we found in the human body. So this was encouraging in the sense that bacteria are not readily developing resistance to these to the encrypted peptides from the human from our bodies. Then, of course, we tested the anti-infective efficacy of the encrypted molecules against different pathogens, including Pseudomonas aeruginosa and E. baumannii. And we can see that both in monotherapy and also in combination therapy, again, illustrating the synergistic capabilities, the potentiating capabilities when you combine cocktails of these agents, and similarly here. And then we also tested their anti-infective efficacy in another mouse model. The previous one that I showed was a skin infection model, and this one is a deep thigh infection model, which is the one that typically the FDA wants to see for any preclinical antimicrobial leads. And once again, we can see in the combination example, in the combination therapy group, we see an increased reduction in the infection with respect to monotherapy and then treated control here. This slide is just to illustrate that at least some of the compounds that we found were actually they seem to have a physiological role in the body, and this for they're taking from the literature. We can see that this is 1, 2, 3, 4 different examples. We can see that these particular peptides are produced at different levels, from low to medium to high, and in different areas of the body, which is rather interesting. And we think that, again, the hypothesis that we have is that a lot of these sequences, these molecules, are having an immunological role, helping us fight off invading pathogens. I'm going to switch gears a little bit now, and I wanted to tell you briefly about another project in collaboration with colleagues over in Barcelona, where we've asked the following question: can we create antibiotics that are autonomous? Typically the antibiotics that we use in the clinic today, they're very passive. If you inject them into the bloodstream, they get carried with the blood with no particular sort of direction, right? They passively just move through whatever medium they're in. And so we partner with colleagues, like I mentioned, over at IBEC, in the institute, and we decided to do the following. We decided to load antibiotics onto a nanomachine that is capable of self-propulsion through space, and in such a way that the nanomachine was able to direct the antibiotics throughout the site of infection, clearing the infection as it moved. And here you will see this in the first quadrant. Here you won't see, you will only see brown M motion. The reason for this is that the substrate that catalyzes the reaction of self-propulsion is not present. But in the other ones, you will see self-propulsion, and you will see how the nanomachine is moving the antibiotics throughout a distance through self-propulsion, and the antibiotics, as they move, they're capable of killing the infection. This is the demonstration in vitro, but we've also in that paper we were able to show this concept in an animal model as well. And of course, this has implications. At least it opens new thinking around how can we have more personalized treatments, more targeted treatments, where the antibiotics go specifically to the site of infection, and they don't have unintended consequences or side effects, killing other types of cells, human cells, or other types of bacteria that might be beneficial for us. And just to sort of wrap up this section, I've told you how we can now use computers, and we're very excited about the capabilities of computers to both be able to design and discover novel antibiotics. And now what we're working on is coupling that to a synthesizer, a machine that will make the molecules the computer tells it to make, and then streamlining that with a screening facility to screen for antimicrobial activity or other types of activity. We'll also have side projects on anti-cancer, for example. And then the key is to build machine learning models to bring that back to the computer, all the functional information, so that we can have a self-learning sort of platform for molecular discovery. So hopefully some of these efforts can someday lead to a drug that can help people. And this is just to illustrate how young this field is. This field at the intersection between AI and antibiotic discovery. This is a retrospective study looking at PubMed results, so publication results, and we can see that the intersection between antibiotics and AI, the publications didn't really, there was almost nothing until really 2018, where the number of publications started increasing. So it's a very young field. I always like to encourage audiences to join it. It's also highly interdisciplinary. In my lab, for example, we have people from computer science, from physics, from biology, from chemistry, all working together. So really encourage those young minds to join these efforts. This is a book that we recently published, published by the American Chemical Society. It's entitled Machine Learning for Drug Discovery. And I just wanted to show it because I think it's a great primer for anybody interested in joining this field and in getting an introduction to this area. And finally, I wanted to just mention briefly some of our efforts during the pandemic around developing diagnostics, low-cost diagnostics. And and here really we were extremely motivated by the huge health inequities that we experience in our society that were, of course, heightened even more during the pandemic. I mean, I still remember that at the beginning when the vaccines were deployed, I think 10 countries purchased 75% of all doses. Even today, we have countries in the world where not even 5% of people have even received the first dose. And so, and similarly with diagnostic tests, many countries do not really have access to accessible, affordable technologies. And we're really moved by all this. And so we, as a team, we got together and we were able to actually develop 3 different biosensor technologies, low-cost biosensor technologies against COVID. And won't go into all the details, but we were able to develop these 3. So the first one was rapid. This is where we published it, the report, and this is the accuracy and in clinical samples, using clinical samples from patients in terms of its ability to detect SARS-CoV-2 in samples, both nasopharyngeal samples and saliva samples. lead was the second one, where we basically developed this test tube, this plastic tube, with 3 pencil leads. We really wanted to develop a technology that was using materials that are highly accessible, and pencil leads are everywhere. And pencil leads, the cool thing is that they're composed of graphite, which is electroconductive. And so we essentially use the pencil leads here as electrodes, and the one in the middle, the working electrode, we functionalized it with ACE2, the receptor for SARS-CoV-2, right, and the spike protein. And so then using electrochemistry, that was the underlying technology that allowed us to detect the binding between the SARS-CoV-2 spike protein and ACE2, and we're able to capture that very, very nicely. So that allowed us to detect it with an accuracy of 87.4, clinical samples. And finally, we developed this technology called Color that is very different from the other 2. It doesn't really use electrochemistry. It's more like it's an optodiagnostic. So think about a pregnancy test where you, through a color shift, you know whether you're pregnant or not. This is similar. So through a color shift, you know whether your sample, your saliva sample, or your nasopharyngeal sample contains the virus or not. And the accuracy was 90% in our tests. And we've been using this free app. This is obviously a qualitative test because it's an optodiagnostic, and through a color shift, you see the result. But we've been using a free app that allows you to quantify pixel intensity, and we can actually quantify that quite nicely as well. And it correlates with PCR results. So just very briefly, but if you're interested, we'd love to follow up on any of these during the Q&A section, or we can follow up via email. And finally, I'd like to obviously thank the team that has allowed to do a lot of team and collaborators that have allowed us to do a lot of the work that I've presented today, and our funders for enabling us to do the work we do. And here I'll leave links to our website, my email address, and Twitter account in case anybody wants to get in touch through those means. I will always love to interact with the community in any way possible. So this is all I have for today, but thank you so much for your attention. Love to take any questions.
Fantastic. Thanks so much, Prof. Cesar. Yes. I do have a few questions, comments, if you have time.
Sure.
So as a clinician, definitely I'm guilty. In the hospital, we treat lots of people that have infections. It could be a chest infection or a urinary infection, for example. I'm a gastroenterologist, so we see a lot of colitis, etc., which is more inflammation as opposed to infection. And then definitely we reach a problem, like you mentioned, that sometimes the antibiotics don't work. So we call the lab, and they say, okay, there's resistance, and you're doing great things in terms of solving that issue. I wanted to ask, firstly, what as an engineer made you very passionate about this particular topic? Because you've got to be passionate to obviously focus on this. And I find people's backgrounds and life stories very interesting. Why did you pick this particular area?
Yeah, it's a great question, Neel. So, I mean, I guess during my undergrad, I remember the first class where I played around with bacteria, and I thought they were fascinating. I remember looking at them through the microscope and trying to learn their intricate sort of lifestyles. And then during my PhD, I did my PhD in microbiology and immunology. So I pursued that passion from a bioengineer sort of perspective. And and then I kept learning about the huge problem of antibiotic resistance and how we really need to think outside of the box to be able to design and discover novel antimicrobials. And I just really, honestly, it's something that is my passion. And but my underlying motivation is always to, hopefully during my lifetime, we can come up with a drug or drugs that can help people, and that really is the fire that drives me every single day.
Yeah, amazing. Yeah, I can see the passion coming through for sure. The other thing I wanted to say is we as clinicians, we get kind of told off. I know it's probably a childish way of saying it. If we prescribe too many antibiotics, so health policy institutes, then the hospitals, depending if you're public and private. If you're private, obviously keep prescribing because they enjoy that. But if you're in a public system, we sometimes have to ration the antibiotics. Do you think the technology that you're focusing on also has potential in this regard, that it'll be able to look at a patient in the future and say, okay, there may be an infection there, but we don't have to suddenly rush in with antibiotics. We can hold off, because as clinicians, we sometimes hesitate. So we feel as a default, we should throw medication at patients straight away rather than the kind of wait thing.
I mean, I think it's essential to diagnose better. I think, for example, when somebody comes in with a cold, that typically, or with some sort of symptoms, cold-like symptoms, that typically is a viral infection. So I think for clinicians to have better diagnostics that they can utilize and be able to identify what the culprit is in each case, I think that's something that we don't currently have. That I think it's preventing clinicians from providing better care, perhaps, or more accurate care to patients. So I think there is definitely a gap there, and I think some of the efforts currently in diagnostics that we saw during the pandemic, those are definitely going to help, I think, in the future, not only for SARS-CoV-2, but hopefully beyond COVID and for other types of viral infections, bacterial infections, and hopefully, out of all those efforts that we've seen and all that work, we can see better technologies that help clinicians diagnose better. I think that's, in the end, the clinician can only do so much if you don't have the tools to be able to help you. So I'm hoping to see more progress along those lines in the near future.
Amazing. We have a comment and also a question. So I'll start with a comment from Mary A. She says, "Thank you very much. I will definitely read the book," which is very nice. And then Alice Korosek, I hopefully pronounced that correctly. "Thank you. Is the existing library of all non-polypeptide molecules already exhausted for possible new antibiotics? New beta-lactams with exotic rings / structure ? So
yeah, great question. I mean, it's probably not all exhausted. I mean, I don't think everybody has sampled every single molecule, but pretty much out of the efforts in the field of people doing bioprospection expeditions going around the world, looking in soil, looking in different areas to try to find organisms that produce antibiotics, that's, for decades we've been unable to find anything new. And so I don't know if that's because it's been completely exhausted, or because we don't have the tools or the technologies to be able to identify new things. It could be one of those two, or maybe there are other options there to explain those failed attempts. But I think not every single molecule has been explored. I mean, I wouldn't claim that. And so there's room for potential discoveries there as well. I mean, I just think that machines, they do help us do the exploration a lot faster and more efficiently than if you have to go, explore the natural world and try to find every single type of molecule out there, So that's kind of the rationale that we're using in the lab. But there are many other groups looking at natural products, and those are all very much viable and great strategies as well. I think the bottom line here is that the more approaches we take to this huge problem, the better. So the more people we have working on different potential solutions, I think the better it will be for humans and for our society.
absolutely, yeah. I think global collaboration is always a good thing. I do have a final question, and then I'm sure you're very busy, so I'll let you go. You mentioned on one of the slides, I think towards the end, about the targeted drug delivery, which I think is a very interesting area. What are the problems that researchers like yourself are facing in this regard? How do we, finally crack it, so to speak? So with inflammation of the bowel, we also are very interested in targeted drug delivery, because we just give medication which, like you mentioned, goes into the bloodstream, and we hope it makes its way to the right place. But we definitely think a targeted approach is needed. What are the limitations at the moment that you're aware of in this area?
Yeah, I mean, as you very well say, I mean, I think most medication, I don't know of any drug that does not cause some sort of side effect or off-target effect. And so in the case of antibiotics, most antibiotics that we have are pretty broad spectrum, in the sense that they kill indiscriminately. They kill pathogens, they kill commensals, which are good bacteria. You're very familiar with them, I'm sure, microbiome members that actually are helpful to our organism. And so I think it's important to also perhaps try to design antibiotics that are targeted, meaning they only kill the bacteria that is causing the infection, and also developing delivery vehicles such as the nanomachines that I mentioned, that can deliver the antibiotic to specifically the target site of infection, and so that it only goes there and it doesn't go all around the body, causing sort of wreaking havoc in other areas of the body. So I think this, more conceptually, this takes us to the more general concept of personalized medicine, and developing medical treatments that are specific to the type of problem or the type of illness or disease that is at hand, and that is not causing sort of unintended unwanted effects.
Fantastic. Yeah, it makes perfect sense. Okay, great. Well, thanks very much for your time. It's a very detailed presentation, very exciting, and you are at the forefront at this field. So very happy that you could join us for our webinar series. And thank you once again. And for those of you that couldn't attend, we will have the recording available on YouTube, so we can continue the debate there.
Thank you so much,
Thanks very
Neel.
much, Prof. Cesar.
It's been a pleasure. Yeah.
Perfect.
Thank you.
Thank you.
Take care.
Take care.
Bye.
Bye-bye. Bye.